Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the gel filtration chromatography, and in the previous lecture we discussed about the basic principle of gel filtration chromatography, how to pack the columns, and then what are the different aspects you have to consider while you are running the gel filtration column or when you are maintaining a gel filtration packed column. So, now in today's lecture we are going to discuss about how you can be able to exploit the gel filtration chromatography to answer some of the questions or scientific problems. So, in this series let us discuss the first problem. So, the such problem what we are planning to discuss related to gel filtration chromatography is where it could be for the scientist or it could be for the PhD students. The basic idea of discussing this problem is that we would like to emphasize that the gel filtration chromatography is a very robust tool to answer many of the complicated questions, and performing a gel filtration chromatography is easy. So, that is why if you would be interested, you could be able to utilize the gel filtration chromatography is as one of the tools to answer these questions. So, let us start with the first question. So, the first question is about a PhD student who wants to determine the structure of a protein X from the Mycobacterium tuberculosis H37 RB. So, Mycobacterium tuberculosis H37 RB is the causative agent for TB or the tuberculosis with the help of the X-ray crystallography. And what he has done? He has cloned the protein in E. coli expression system, and then he has purified the protein and set up the crystals, but what he could get is he was getting the precipitate instead of getting the crystals. So, in a typical X-ray crystallography related structural determinations, what you are supposed to do is you have to first isolate the proteins in a large quantity in a purified form. So, what you are going to do is first you are going to get the purified protein, then what you have to do, you have to concentrate this protein so that it should reach to a concentration where it will start showing the crystallization, and then you are incubate this in a different conditions or different crystallization conditions, and at the end in any of these conditions the protein is going to get precipitate in a regulated fashions, and as a result the protein is going to crystallize and give you the crystals. But with this student who has actually been interested to solve the structure of the protein X using the X-ray crystallography, he has cloned the protein. So, one of the easiest way of getting the purified protein is that you clone the protein in any of the overexpression systems such as E. coli or baculo or mammalian expression system, depending on the yield and the protein of the choice. And then he has performed, then he incubated the protein, the concentrated protein in a different conditions, and what he could found is that he, instead of getting the crystal, he was getting the precipitate, which means the precipitation of the protein was very fast compared to the very slow, so that he should get the crystal. Now, what the first thing he want to ask is whether the protein what he has purified from the E. coli is good enough, or it is in a good native conditions, or the protein, the quality of the protein what he has isolated from the E. coli is good, so that he could actually be able to verify the other parameters. So, what he wants to design the few experiments to check the quality of the protein purified from the E. coli system. So, what do you mean by what we mean by the quality of the protein is that he would like to determine the molecular weight of the protein and then he wants to also ask whether the protein is present in the native conformation and what are the different oligomeric status of this particular protein. So, he wants to ask 3 questions: whether the protein is properly folded, means the protein is maintaining a 3-dimensional conformations. Number 2, what is the molecular weight of the protein, and number 3, what are the different conformational status of this particular protein. You know, the protein is being produced as a monomer, but these monomers comes together to give you the dimer or tetramer or even the oligomers, and there are many proteins which are actually being present either as a monomer, dimer or the higher oligomers. So, to answer these questions, he has to design an experiment related to gel filtration, but based on the basic principle that the distribution coefficient is equivalent to the V e minus V o by the V I, which means if you would like to utilize the gel filtration to determine the molecular weight should be in proportion to the radius of gyrations. So, the molecular weight and the size of the protein is related to the shape of the molecule, and the relationship between the molecular weight and the radius of gyration is Rg is directly proportional to the Ma, where A is a constant and it depends on the shape of the molecule. A is 1 for the rod, which means the Rg is directly proportional to the molecular weight. A is 0.5 for coils and A is 0.33 for the spherical molecules. So, this means that the molecular weight is directly proportional to the radius of gyrations, and because of this relationship you can be able to utilize the gel filtration to determine the molecular weight of this particular protein, and also you can be able to determine the oligomeric status as well as the whether the protein is present in the native conformation. So, what is the experimental design? For the experimental design, the first thing what you have to do is you have to run the multiple proteins of the different molecular weight. So, in this case, for example, we are running the different types of molecular weight proteins, and then you have to draw a calibration curve between the KAV versus the log molecular weight, and that is how it is that calibration curve can be used to determine the molecular weight of the unknown protein X, as well as the oligomeric status, as well as the whether the protein is present in the native conformation or not. To perform these experiments, what you are supposed to do? You are supposed to do a gel filtration chromatography, where you are going to first run the standard proteins to draw a calibration curve, which means you have to determine the relationship between the KAV versus the log molecular weight, and once you know that relationship, you can be able to run your native proteins or the protein of the protein X. So, suppose the protein X is giving you the KAV value of this, that actually can be used with the help of the calibration curve to determine the molecular weight. Apart from that, you can also run the protein X into the SDS page, and that actually will give you the denatured molecular weight, whereas the gel filtration chromatography is going to give you the native molecular weight, which means what is mean by native molecular weight means is if the protein is a dimer, it is actually going to give you the dimeric molecular weight, whereas the denatured molecular weight means it is actually going to give you the molecular weight for the monomer. Now, utilizing these two tools, the gel filtration chromatography and the SDS page, you can be able to determine the oligomeric status. So, how to determine the oligomeric status of the protein? First, you have calibrated the column, then you have drawn the calibration curve. Now, you have the native molecular weight, which you are going to determine with the help of the calibration curve, and then you are going to determine the denatured molecular weight with the help of the SDS page, and that if you put it into the formula, that is the oligomeric status is equivalent to the molecular weight you are going to get from the gel filtration divided by the molecular weight, which is going to be get by the SDS page. Apart from that, the quality testing can be also done when you are going to analyze the protein X onto the gel filtration column. What you are going to get is you are going to get the pattern of protein X in that particular buffer conditions. So, you can imagine that if I have, you can have the protein X either giving you single peak, or you can have the protein X, or in a different conditions you can imagine that the protein X might be giving you 2 peaks. Okay. So, then what you can do is you can determine the molecular weight of the peak number 1, you can determine the molecular weight of protein number 2, and if the peak number 1 is of the double the size or higher molecular weight size, that means that the protein X is probably be present in the 2 oligomeric status, which means either it could be present as the dimer or the monomer or the mixture of both, and that is very common in many of the proteins, that the proteins are present in a dimeric conditions as well as the monomeric conditions. And when you have such situations and you are interested to study this protein, it is always important and always recommended that you actually isolate these individual peaks, so that you will be able to study the dimer versus monomer separately, and that any of either of these molecules or either of these oligomeric status of the proteins might give you the crystals or might precipitate as per the very slowly, and it will give you the crystals. So, that is also one of the strategies what people also plan and try, and that actually gives you the successful crystallization of a protein, and it actually eventually give you the structure of that particular protein. Now, let us move on to the next problem, and the next problem is that the protein X is present in 3 oligomeric status. So, here you already know that the protein X is present in 3 oligomeric status, one is monomer, dimer and tetramer, which means the protein is present in 3 oligomeric status. This is the tetramer, this is the dimer and this is the monomer. Okay. So, it is already been set that the protein X what you have synthesized or what you have produced having the 3 oligomeric status, and now the scientists want to study the stability of this particular protein. So, before get into the detail of the stability of the protein, because the stability is a very vague term and you can actually have the stability of a protein against multiple parameters. For example, you can have the stability of a protein against the protease treatment, you can have the stability of a protein against the denaturation conditions such as the urea, GDMCl and all other conditions, and you can have the stability of a protein against the thermal denaturation. For example, you can incubate the protein at different temperatures irrespective of the conditions except the protease. If you are treating a protein with the denaturating agents, or if you are treating a protein with a biophysical parameter such as the temperature or other kind of parameters, it is actually going through a different discrete steps in which the protein, which is 3 dimensional conformations, is going to denature and going to adopt extended conformations. So, in that process the protein is going to be give you the multiple conformations. For example, you start with the native conformation which is actually 3 dimensionally folded protein. So, that is actually a compact structure. It will be where the protein is going to actually arrange all his amino acids around a center, but as soon as you apply the small amount of urea, the protein is going to be partially folded, and as you can see the diameter of this protein is now increasing, because you are breaking the interaction between the side chains and even the main chain also, and because of that it is actually adopting those extended conformations and the structure is now opening. So, that is the diameter of the molecule is also increasing. Now, if you further denature with the help of slightly more concentration of urea, what will happen is it will further going to break more and more interactions, and ultimately it is going to increase its diameter, and ultimately when you have a very high concentration of urea such as the 8 molar urea, the protein is going to be in a extended conformation, and that is how it is actually going to acquire the very high molecular hydrodynamic surface area or the diameter. Now, if you would like to follow this kind of changes and you would like to use that as a way to answer whether the protein is stable or not, what you can do is you can take this protein X and you can perform a gel filtration chromatography to answer these questions. So, in the experimental design, what you are going to see is, if you are having all these conformations and if you are going to analyze them onto the gel filtration chromum, what will happen is, this is the largest size, this is the middle size, this is even smaller than this, and this is the smallest among them. So, if you follow this, what you will see is that the unfolded protein, which is completely unfolded protein, is going to be present in the wide volume, whereas the other 3 conformations like the 3, 2 and 1. So, 1 is going to be elute as per its native conformation. So, as per the native positions, the 2 and 3 are going to give you the intermediate positions, and the 4, which is actually the unfolded protein, is going to be present in a extended conformations. So, that will be completely excluded from the column and it will be present in the wide volume. So, that is the way you can be able to design, the experiment and can be asked the answer that whether the protein is more stable or less stable, because the amount of urea what you are going to use in this experiment is going to determine how much urea is going to require to bring the 1 position to 2 position or 2 to 3 position or from 3 to 4 positions. The amount of urea what you are going to use is actually going to indirectly tell you the how much stable the native conformation is, and that question can be even asked even for the different oligomers, because what you can do is, if you run this protein on a gel filtration column, you are going to get 3 peaks, which is actually the tetrameric peak, dimeric peak and the monomeric peak. What you can do is, you can isolate these peaks individually and then you can be able to perform the similar experiments along with the individual oligomeric status, and that actually will tell you which oligomer is more stable and which oligomer is less stable. So, in the experimental performance, what you are supposed to do, the protein is incubated with different concentration of urea, which means from 0 to 8 molar urea for 8 to 10 hours at 37 degrees Celsius, and in parallel you have to run a gel filtration column which is equilibrated with a buffer containing urea, same as an incubation buffer, which means if you are running the 1 molar urea and you incubated the protein in 1 molar urea, that buffer also should contain 1 molar urea, so that while the protein is also running through the column, it should not experience any kind of reversible folding, because you know that if you will provide the non-denaturating conditions and if you provide the non-denaturating conditions, the protein will tend to fold. So, that is how it is actually not going to give you the real picture. So, what you are supposed to do is, you have to use the same concentration of urea for equilibration of the buffer and the same urea in which you are going to incubate the protein, and then you analyze the results. As the concentration of the denaturing agent is increasing, the protein will unfold with an increase in hydrodynamic surface area. As a result, the protein peak will shift towards left, at highest concentration of denaturant, the protein unfolds completely and the mostly appear in the wide volume. So, what you are supposed to do? You are going to do a gel filtration chromatography with the buffer containing urea, and you also have to incubate your protein with the different concentration of urea, which means from 0 to 8 molar urea, here also 0 to 8 molar urea. Now, how the results will come? The results will come like this, where you are actually initially going to see a peak of the native conformation. Now, this is at 0 molar urea. Once you increase the urea concentration, what will happen? It is actually going to shift and going to give you the two peaks. One peak which is for the native protein, the second peak which is for the partially denatured protein. Now, as you increase, so suppose this is at 2 molar urea. Now, if you increase the further, what will happen? You are going to see one more peak, you are going to see one more peak, and you are going to see that the native protein is now reducing in concentrations, and you are getting the partially denatured protein which is of 2 molar urea, and you are getting another peak which is actually corresponding to the 4 molar urea. And now, if you further increase, you are going to get very high peak and probably native peak if there is a still the protein which present in the native conditions. Otherwise, all the protein is going to be get converted into the denatured protein. So, that is how, if you can see this movement, you can be able to conclude that the protein is showing you a denaturation curve, and what will be the stability of the protein, because the mutual proportion of these conformations in different peaks is going to give you a idea how much the protein is stable. Because even at 8 molar, suppose you have the 20% protein still left in the native conformation, which means this protein is very stable, and probably it is can withstand the very harsh denaturating conditions. Now, let us move on to the third problem, and the third problem is that a scientist has isolated a unique protein responsible for induction of apoptosis in the cell. He suspects that the protein might be interacting with DNA and disturb its replication. Now, he needs to design experiment to study the interaction of DNA with the protein. So, what he wants, he has isolated a protein P which is actually interacting with DNA, okay, and that is how it is actually interfering or disturbing the replication of the organism. And once you are interfering the replication of the organism, the genomic DNA of that particular organism cannot be synthesized. So, as a result, the cell will have no option but to go for the apoptotic pathway, where the cell is going to stop its growth and it will go through the death pathway. So, ultimately it will go for the apoptosis. Now, if I have to design an experiment and I would like to answer these questions, if you remember when we were discussing about the ion exchange chromatography, that time also we have discussed how you can be able to study the interaction of the DNA with the protein, where we have taken an example of a transcription factor and the promoter region present on the gene. So, in this case also you can be able to answer the similar questions, or you can be able to explore the similar questions also with the help of the gel filtration chromatography. Now, what you are supposed to do in your experimental design, what is the experimental design? Where you are actually going to incubate the DNA. So, when the ligand or the DNA binds to the protein, it induces the conformational changes into the protein, and also it increases the molecular weight. So, when the protein is going to interact with DNA, it actually going to affect the first the shape, the size of the protein. The second, the protein DNA conformation is going to be of higher molecular weight, which means you are actually going to study this simply by two ways. One, if the conformation of the protein is going to be changed, it is actually going to change its position on the gel filtration curve. So, that is what you see. If you are injecting the native protein, it will actually going to be present at a different position, but if the ligand and the protein is present, it is actually going to give you the high molecular weight, and that is how it is actually going to show you the separate peak. So, results into the change in the size or the shape. In addition, ligand is small in size, whereas the protein ligand complex is big and may appear at a distinct place in the column. So, this experiment we are showing you only for the DNA, but you can be able to answer even the interaction of the small ligands such as the substrate with the protein also utilizing the gel filtration chromatography, because the substrates are very small, they are going to be run as separate peak, and when they are in a complex with the protein, they are actually going to run very separately from the ligand peak, and that is how you can be able to understand the interaction of the ligand with the protein. Also, the amount of the ligand present in the condition B versus the amount of the ligand present in the condition A can be used to ask how much ligand is being consumed to give you the ligand protein complexes. So, if you follow that, the ultimately what will happen is you are only going to get the ligand protein complexes, but there will be no ligand present, and that is the status where all the ligand is being consumed to form the protein ligand complex, and that is the concentration can be used even to calculate the dissociation constant and all other parameters. How to perform these experiments? You perform these experiments, and what you require? You need a gel filtration chromatography where you are going to incubate the gel filtration buffers containing the DNA, and you have to incubate the protein with different concentration of DNA. Okay. So, what you have to do in the step 1, a gel filtration column is equilibrated with the buffer and the elution profile of the ligand is recorded. Now, the column is equilibrated with the buffer, okay, containing the ligand molecule, or if the ligand molecule is big, for example, the DNA, then you do not need to do equilibration of the column with the ligands, then you can directly equilibrate the column with the buffer, and you can run the DNA separately, you can run the protein separately, and then you can put them together and see where they are actually eluting in these conditions. So, as the concentration of the ligand is increased, the protein binds the ligand and form a larger complex with an increase in hydrodynamic surface area. As a result, the protein peak shoots towards the left. So, in this case, what you are supposed to do is you are going to run the 3 gel filtration column, first for DNA. So, DNA is very large, it will going to give you a peak. The second, the protein. So, protein is also going to give you a peak. So, this is for protein, and then for the DNA protein. So, in this case, you are going to get a intermediate peak, which is because here you are monitoring the peak at 260, here you are monitoring at 280, and here also you are going to monitor the protein, not the DNA. So, as a result, you are going to get a intermediate peak, and that actually is going to be corresponding to the DNA protein complex. Now, the question is, if you get a change in the peak of the protein, that could be specific, that could be non-specific, because the protein for example, especially in the case of DNA, which is actually a molecule with a very diversified charges, and it has a basis. So, it actually provides lot of positive and negative charges, it provides the hydrophobic cores. So, there are possibility that the protein might interact with the DNA non-specific as well. So, to confirm this, what you have to do is you have to do a validation experiments to confirm this. Now, what is the results you are going to get and how to validate that is that in the absence of the in the absence of protein, the ligand is going to run separate, and when you are putting a protein, the ligand is going to form a complex with protein and going to run a separate peak. But how to verify whether the protein DNA complex is a real complex or to a non-specific complex, you have to do a verification experiments. Now, how to do a validation experiments? Now, validation experiment can be done in two ways. One, where you can just remove the DNA, okay, because the protein is forming a complex plus DNA to give you the protein DNA complex. Now, if I remove this, okay, so if I remove this, I should not get the PD complex, okay, which means if I am seeing a peak which is intermediate peak, okay, because this is the DNA peak, this is the protein peak. So, this is the DNA peak, this is the protein DNA complex peak, and this is the protein peak. A shift in the peak could be because the DNA is interacting with the protein, a shift could be because some kind of artifacts while you are running the second time. So, to understand that, what you do is you take this complex, isolate it, and then you degrade the DNA and run it again. If you run it again, it should give you a peak at the protein positions. If that happens, that actually indirectly will verify that the shift in the peak is because of the DNA. The second thing is, because you have isolated the complex, you can also analyze this complex onto a agarose gel, and that actually will give you a band for the DNA which you have used for the interaction studies. This is the one approach. The second approach is that because the DNA protein interaction is whether it is specific or non-specific, to answer this, what you can do is you can simply do a mutational studies. So, what you can do is you can just generate the mutated protein, and that mutated protein should lost the binding of the protein to give you the complex, and it should also run at the the native conformations. So, this is all about the designing an experiment to study the protein DNA interactions. We have also discussed the similar type of experiment when we were doing the ion exchange chromatography, and with this let us move on to the next experiments. The next experiment is, or the problem third is, that the scientist wants to record a CD spectra in water, but the protein is being purified with the differential precipitation of ammonium sulphate and has large amount of ammonium sulphate. The protein is sensitive and degrades during long hour of dialysis. So, he has to design an experiment to remove the ammonium sulphate from the protein. So, what is mean by is that you have a cell lysate, okay. This cell lysate they have incubated with ammonium sulphate. So, what happen is that actually has given multiple peaks, and out of these multiple peaks, one of the peak was containing the protein of your interest. But the issue is, and this for this protein he wants to collect a circular dichroism spectrum, but this protein contains large quantity of ammonium sulphate. So, if you contain, or if the protein contains the large quantity of ammonium sulphate, it actually is going to interfere with the it is actually going to interfere with the further analysis and getting the proper CD spectrum. So, what you have to do is you have to do a desalting or the removal of the small molecule from the protein is important for activity assay and other downstream processing. So, what you do is you load this protein which is having a complex with the ammonium sulphate, and if you run it on a gel filtration, the protein is going to be run separate and the salt is going to be run separate. And you can be able to do that simply by running performing the chromatography in a very small column. So, one of the column which is called as the NAP10 column. So, these are the commercially available NAP10 column which you can use to desalt the protein. You can remove any ligand which is of a smaller size. So, when you load the desalting column onto the NAP10, so you can get the protein separate and the salt separate, because what happen is a gel filtration column is equilibrated with the buffer of water, okay, and then the sample for desalting is loaded. After the run, the protein and the salts are eluted separately as a peak. We have prepared a small movie clip with the NAP10 desalting column, and with the help of this movie or the demo, you will be able to understand more nicely how to perform the desalting of a protein sample, whether it contains ammonium sulphate or other kind of small molecules, and how you can get rid of this. Today we are going to give you a demo about how to desalt the protein using the NAP10 gel filtration column. So, this is a typical NAP10 gel filtration column. What you see here is the packed column, and this is a pre-packed column what we have purchased from the company, and it is being preserved in the buffer containing 0.2% azide, because the NAP10 columns are mostly sensitive for the organic solvents. So, that is why it is recommended that you preserve them in a buffer containing the azide. So, now first we have to do is, what we have to do, we have to equilibrate this column with the solvent in which you are interested to do the buffer exchange. So, if in this case we are actually trying to desalt the proteins, so what we are going to do is we are going to equilibrate this column with the water, and then we are going to load the protein with the help of the buffer itself, and then we are going to load the protein into this column, and then we are going to start collecting the fractions, and then that is how it is actually going to remove the salt from the protein. In case you are interested to keep your protein in some buffer condition, for example, if it is not very stable in water or non buffer conditions, then you can equilibrate the column with a particular buffer. So, let us start the NAP10 column mediated desalting. So, what you have to do is, first you have to fix your column into the stand, then you have to remove the cap from the top, okay, and then you remove the lower end, and then you let the column to run, and you see this level should come to the level of the disc, and then you can start pouring the buffer of your choice. So, this is the this time we are running the column with the help of only the manual mode. If you are interested, you can also connect this column to a purification machine also, but since this only takes 5 to 10 minutes, people normally don't prefer to add the connect the column to the purification system. So, what you see is now the buffer is reached to the disc, and now what we can do is we can simply add the buffer or the water and fill it up to the top. So, while you are adding, you have to ensure that there is a smooth flow of buffer from the lower end of the column, and also there is no particulate matter present onto the top of this top filter, because that top filter is being kept simply to avoid the the exposure of the column material to the particulate matter or the aggregated proteins. So, that's why the it actually protects the column from getting any kind of damages. So let's first do the equilibration, and then we are going to load the sample, and then we are going to tell you the further downstream procedures. So now what we have done, we have equilibrated the column with the 4 to 5 column volume of the water or the buffer in which you are want to do the buffer exchange of your protein. So what you see is now the level is going to the disk, and in between what you have to do is you have to prepare the sample. So in this case what we have is we have the 0.5 ml of the protein. So whatever the protein solution you have to bring the protein solution to 1.5 ml. So what you can do is just simply add the water or the buffer, whatever you are interested. So add it so that it is going to be 1.5 ml, and then you can just load that. So you can see the level is reached to the disk, and now what we can do is you can simply mix the solution first, and then you load it onto the protein, onto the column. Okay, and you have to wait for this protein solution to get into the column, and if you are interested, you can be able to preserve some amount of sample so that you can verify what will be your recovery. And once this will go down to the level of the column, we are going to add the buffer or the water. So now it is reached to this, and now what you can do is just simply fill this buffer with the column with your water, and you have to fill this up to the top, and then you start collecting the fractions. And what you can do is you can just simply collect the 0.5 ml fractions, and your protein will come out into this 0.5 ml fraction. So what you can do is you can simply collect the 6.5 ml fractions, and that actually is going to, so what you can do is you can collect the 6.5 ml fractions. So I have collected the fraction number 2, and the similar way you can collect the another 4 fractions. And because the protein is going to be present into the exclusion limit, because the protein what you are adding is going to be very big compared to the salt what is being present in this particular protein. So what will happen is the protein is going to be in the exclusion limit of the column, so the protein will come out the first. And if you collect these fractions and analyze onto the SDS page, what you will see is that the initial 3 fractions are going to have the protein of your interest or the maximum amount, where in which the fraction number 2 is going to have the maximum amount of protein, and fraction 1 and 3 are going to have the lower amount of protein. And from fraction 4, 5 and 6 you are going to get the salt. So this is all about the desalting of the proteins with the help of the naphthalene column. So ideally what we are doing, we are just loading a protein salt complex onto a naphthalene column. Naphthalene column is a G25 column, and ultimately you are going to get a protein because the protein is present in the exclusion limit of that column, and because it is very big, so it is actually not going to fractionate the protein, and it will come into the exclusion limit, and as a result the protein will come first and you can collect the proteins. So with this we have discussed about the gel filtration chromatography and how you can be able to utilize the gel filtration chromatography for addressing the different types of scientific problems. And now subsequently in the next lectures we are going to discuss about the affinity chromatography and how you can be able to utilize the affinity chromatography for performing the different types of experiments or solving the biological problems. With this I would like to conclude our lecture here. Thank you.